That's not right. Good morning, everybody. As you can see, this room is slightly less appropriate for filming. Could you just sign your name on this as it goes around so that I know you're here? It's a makeshift register. So because of the filming issues here, there will be occasional times when I'll need to stand on this chair, like so, in order to get myself in the image. I guess this increases your engagement because you're slightly anxious about me falling off. I've got to admit that I'm slightly anxious about falling off as well. On the other hand, you think about the number of YouTube hits I'll get if I break my neck. Okay, so we are broadcasting live, so please bear that in mind if you ask questions. I have to say that. Also today, I would like to talk a little bit more slowly because I think I went a little too fast last time. Two clues I have to this are the fact that some people said we didn't understand everything because you went too fast. The other thing was I finished about 3/4 of an hour early. So I think those two pieces of evidence combined mean that I need to speak a little bit more slowly. So if you want me to slow down, just do that with your hand and I'll know what you're saying, okay? Don't do that, I'll ask you a question then. But if you do that, then I'll know I need to slow down. Okay, so today we're going to be looking at, or continuing really, our little journey into the brain to find out what happens when we learn. And this is going to be very difficult. I'm going to actually begin with a review of what I'm going to talk about, which I could go back to at the end. So this is like an executive summary, because I think it's quite important that you join together some of the things that I'm going to talk about and understand why I'm talking about them. And so I want you just to be aware what the basic facts are that I'm trying to get across here. I've got a feeling that when I press this laser, the slide goes forward. Oh, it doesn't. Oh, good. It was yesterday. Okay, so learning in the brain review. So learning is often synonymous with memory in cognitive neuroscience. Now that's an aberration if you're an educational researcher, okay, because learning clearly occurs in all sorts of ways. We need to understand, we need to be able to apply, we need to be able to analyze, evaluate. But in cognitive neuroscience, learning is very often just memory, so bear that in mind. Perhaps even more radical in biology, it tends to refer to any type of plasticity, any change in brain structure, connectivity or activity in relation to something that's happened in the environment, that is learning. Okay, so it's like a biological definition. So just be aware of the fact that we're talking about learning in terms of cognitive neuroscience, and many of you have an interest in education. In fact, I think all of you do. And so we have two different ideas about learning, at least, or maybe three different ideas about learning: cognitive neuroscience, biology, and education. So be aware that we're bringing together perspectives that are not always discussed in the same lecture. I'm going to be telling you that all animals learn, and that useful learning can be achieved by quite small networks. Okay, so actually you can see something very similar to what you might think was learning with just a handful of neurons. But in order for that to happen, the neurons in the network have to be able to change the efficiency of their connections in relation to something that's happening in the environment. And actually the type of change we're after is what you see in a Hebbian synapse, and I will be explaining what a Hebbian synapse is. But I've put a little definition here. It means these Hebbian synapses change their efficiency in response to simultaneous pre- and post-excitation of the neuron. So if the neuron is already firing when you excite it, then the connection between the two neurons, the one that's doing the exciting and the one that's already firing, will increase its efficiency. That's a Hebbian synapse. Biological connections between neurons are called synapses, but the ones that we're most interested in are chemical synapses. There are electrical synapses that you come across in the reflex system, but the ones that we're interested in are the more sophisticated types, the chemical synapses. We are going to be talking about this change in efficiency in biological terms, and to do that we'll introduce this process called long-term potentiation. That's the biological basis for the Hebbian synapse, and that should be a full stop there. So working memory may also be due to persistent neuronal firing. Okay, so we'll also be talking a little bit about working memory, but working memory is a little different to long-term memory. Okay, so, but I thought we'd throw it in, why not? Memory and working memory are distributed in the brain. So you don't have a box in your brain where you put your memories. A memory is distributed across the brain. I'm going to mention evolutionary psychology, and that is something that I'm throwing in that I wouldn't normally throw in, but actually it's important for those people who are doing the BPS pathway. So I'm going to mention something about evolutionary psychology. I'm becoming more and more skeptical of evolutionary psychology, and I don't care if the evolutionary psychologists are listening or watching this lecture. I thought it used to think it was really fascinating stuff, and it is really enticing, but actually, I don't know. We will mention neuroplasticity. I mean, that is really what we're talking about up here, neuroplasticity, the way in which the brain changes in terms of structure, connectivity, and function. And really just to emphasize that it is the structure that changes as well, and that's quite a big deal. It's quite incredible to think that the different shape of the parts, different parts of the brain, the shape of the different parts of the brain change in response to learning. So as a result of this unit, one part of your brain, at least, probably, or maybe a few parts of your brain will be bigger. Maybe that, of course, that means some have to be smaller, but there you go. I don't know which way this will be. So let's start off with maybe the simplest type of conditioning you can think of. And this was, I suppose I thought I'd start with Pavlov's dogs. And you probably know the story of Pavlov's dogs, that he noticed that you know, when the dogs, well, you've probably seen this yourself, dogs only have to hear something that they associate with food and they get terribly excited and start salivating. So they've learned that that sound of the cupboard opening or whatever, or the cat with a tin opener, you know, the cats and tin openers tend to respond quite well, don't they? And they hear those tin openers going. They know that food is on the way. They've learned something. So yes, animals learn. So it might be good to have a little look at this, hopefully working demonstration of this phenomenon. And I've put the credit on there so that I don't get into too much trouble doing this. And the idea is, do we have any sound, I wonder? Probably not. The idea is that you have this, you could try this at home. Okay, and essentially what you have to do is make sure that you introduce things at the right time. I want to make this dog drool. So I've got Sam. We have a sleeping dog. What I'm going to do is put some sausages there. Probably wasn't. I should have probably put. I probably should have made the noise before I put the sausages in. Actually, I made a mistake there. Can we start again? Okay. Oh no, it was there. I'm not sure if I was fast enough. You need to do these things together, you know. Okay, let it go back to sleep. Yeah. Notice that the dog isn't salivating yet. It's waking up, but it's not salivating when I. Oh, I made a mistake. Oh, you should have stopped me. I'm useless at doing experiments. I'm going to have to carry on a bit longer now. Back to sleep, back to sleep. Oh. Look, nice, he's getting the idea now. You can see inside its brain. Oh, and I've got a diploma, thank you very much. Thank you. Well, it's nothing really. Okay, so you can try that at home. Basically, that dog has learnt to associate the sound of that trumpet. I mean, if I could have shown you that if I rang the bell, it wouldn't have really got very excited. But when you start doing the trumpet now, it will actually start salivating on demand. So how does that actually, how does that happen? Well, we often see animals making, here I go again, reasonable decisions, what appear to be quite reasonable decisions. It seems to know, it seems to have learned something. And a lot of this can be explained in terms of conditioning. So, you're actually, you're associating two things together. You've got your basically an unconditioned stimulus here, which may be the light bulb like that, and then you put out some food, and then after a while, you see that when you just switch on the light bulb, the animal starts behaving as if it's going to be eating, or so start salivating. That's what we see. But actually, you get all sorts of different types of conditioning. All sorts of different types of what we call classical conditioning, this very simple type of conditioning. And it can become quite complicated. I mean, for example, this second order conditioning, you can actually associate two things together. And then when, if this has already become, if this light bulb, for example, has already become associated with food, and then you associate the light bulb with the sound, then when you hear the sound, the animal will start salivating, as if it knows that the sound is associated with the light bulb and the light is associated with the food. So you can get quite complicated effects, and there's a whole sort of list of them here which you can work out for yourself. And this helps explain some interesting phenomena that you may have come across. Have you ever heard of Clever Hans, the counting horse? Clever Hans could actually, if his owner sort of held up so many fingers or whatever, then the Clever Hans would actually nod his head, and I think he could do simple sums as well, and he could nod his head for the right answer to actually explain what the right answer was. So here we have a horse that appears to be able to count, and I mean, in its day, Clever Hans was a phenomenon and became very famous, but ultimately, a psychologist was able to not just explain, but actually provide evidence for the fact that this was just a case of classical conditioning, that the horse had begun to realize, not from the hand being held up or from the number of carrots or whatever, but actually it could actually detect, it had become conditioned to respond to the body gestures of the audience around, and particularly the owner. So Clever Hans became very attuned to the body language of its owner and knew when it was supposed to respond. So by simply going, do you mean 1, 2, 3, 4? And if the answer was 5, the horse could tell that the owner was becoming excited, and it associated that excitement in the body of the owner with the fact that it would be rewarded soon with some food if it actually started nodding its head. And this is how it worked. So you can explain some apparently clever animal behavior purely in terms of classical conditioning, but you have to say that actually animals can appear much more sophisticated than that simple conditioning response, that classical conditioning response. And you can train, as you know, sheepdogs to do quite wonderful feats of work in terms of rounding up sheep, and their behavior appears very sophisticated in terms of the training that they've received. And this has to be explained in terms of a different type of conditioning, whereby you are actually anticipating the consequence of what you do in terms of the rewards and the punishments. And we call that instrumental conditioning or operant conditioning. Not only that, but it also requires quite often gaining some sort of cognitive map of the options that are available to you. And perhaps a clearer example of that is the way in which rats that get to know a particular maze, if you block off one section of this maze, the route that it has normally taken to get to the food, then it can work out what the second best route is, or the most likely route to the food. So it's not simply remembering left, right, left, left, right. It actually has some sort of construction of the entire maze inside its head. So these sorts of learning situations appear quite impressive. And so the question we have to ask, without falling off the chair, is how would neurons have to behave to produce such behavior? How can they make these sorts of educated guesses in new situations from such a cognitive map? Now, I introduced this image before. We have a network of neurons, and I said that if you got some input here, maybe you've got your cognitive map here, and then that can actually produce outputs here. And actually, even this type of neural network is capable of some sort of guesswork. So it has what we call graceful degradation. So if in fact the input is imperfect in some way, then it can make a pretty good guess at what it might mean still. So this is the difference between a parallel network here, where you've got input and output going through, a whole layers of, so you've got 1, 2, 3 layers of thinking. It's very different to a computer where you actually have things working in series. So a computer's way of thinking, you can often summarize in terms of going from one box to another box in a flow chart. That's serial thinking. But in neural networks, you often have inputs, and actually chains of events going on at once. So we could have another neuron connecting those two. So there'd be information going down there, and there'd be information going down there. So we have parallel processing. And there's usually some sort of redundancy in that parallel processing. So if you actually break a connection, or if the input is imperfect, then you still get some sort of output which appears reasonable. Now, if I go into your computer and I remove a component, or if I basically take the code in your computer and I just change, I don't know if you've ever done any computer coding, but this is so frustrating. You just leave a full stop out of your computer code, the whole thing will not work. Okay, so that is because it all runs on serial processing. There's a massive flow chart, a sequence of events that has to follow through in order to get an output. Parallel processing is not quite like that. It's more there's more redundancy built in. It can make guesses. Now, that's all very well, but it all relies on the efficiency of these neurons being, these connections being just right, so that information that comes in here is transformed into just the right form of information on the outside. So, if I showed you that this network did in fact do the job, so that the input here produces the right output there, you still have to ask the question, well, how did it actually get to that point? How did it manage to change, if this neural network is learning all on its own, how did it manage to change the efficiency of these synapses so that it could actually learn? How did it end up with the right sort of connections, in other words? I mean, this is an example of a neural network. And you've got input here and you've got output there, and you can see that they're all interconnected. So this is a simulation. This is the sort of thing people do on computers to simulate neural networks. And the odd thing is that if you put in a hidden network like this, it allows so much more flexibility in terms of the way the input can be mapped to the output. The potential intelligence, if you like, of the network is really increased, and it can do much more sophisticated jobs. So these hidden networks, these hidden layers are actually very important to the intelligence of the network. And an artificial neural network like this, you will see following the same sort of learning curve as similar to a human or an animal. So it's very rapid at first, and then it reaches some sort of level and levels off. Okay, so I think we better talk about what we mean by a biological connection, because we haven't really talked about that yet. A biological connection between two neurons is called a synapse. So this is what you've got in between your presynaptic terminal and the dendrite of another neuron. Okay, so what I'm drawing here is basically that bit there. Okay, you've got your presynaptic terminal going on to the dendrite of another neuron. It's a connection, only we call it a synapse. Now somehow the action potential that you've seen flowing down the neuron in the previous lecture, that has to find some way of getting across to this new neuron, to this other neuron. And actually it doesn't do it electrically. Well, not in a chemical synapse anyway. There are electrical synapses, but they tend to be more about reflex systems. And we're interested in the more intelligent, if you like, synapses. So we're going to concentrate on the chemical synapses. And what happens here is that you basically have your action potential coming down here. And when it reaches the synapse, it depolarizes this membrane, allowing calcium ions to flow through. And when those calcium ions flow through, they essentially activate proteins attached to these vesicles inside the presynaptic terminal. And these vesicles contain neurotransmitters, a particular type of neurotransmitter. There are many different types of neurotransmitter, and they all do different things and play different roles in the brain. An important thing to remember is that the same neurotransmitter can easily do different things in different regions of the brain. Okay, so it's not like dopamine has one role in the brain. It's not like serotonin has one role in the brain. These are all, glutamate, these are all neurotransmitters. Anyway, these neurotransmitters to begin with are bundled up inside these vesicles. The calcium ions come through, they attach themselves to they activate proteins on the surface of the vesicle. That causes the vesicle to merge with the membrane of the presynaptic terminal, and it opens and the neurotransmitters come out, and some of them just flow around and they sort of get lost, but some of them bind themselves to receptors in the postsynaptic membrane here. And that's important because once these receptors become activated, then there's an activation of the postsynaptic cell here, and the way in which it activates, what actually happens next depends on the type of receptor and the neurotransmitter. Okay, so if it's, for example, a dopamine neurotransmitter receptor, then there's a good chance you'll receive some sort of activation. Serotonin tends to be an inhibitor. But let's imagine that there's an excitatory response, in which case it increases the likelihood that this postsynaptic neuron is going to fire. In other words, we have transmitted an action potential here to an action potential in the next neuron. Okay, that's how a chemical synapse works. And these sorts of, that name there is worth remembering, a receptor, these voltage-gated calcium channels, these are the sorts of things that might come up in your little exam. Now, we still haven't explained actually how the efficiency of that transfer changes. And that's the thing which is actually going to make our network intelligent. So when an action potential in the presynaptic neuron reaches a synapse, it causes release of neurotransmitter. This influences the postsynaptic neuron, and that may fire also. So that's what we've just been talking about, how that happens. Now, Donald Hebb thought all about this back in 1949, before we even knew about the biological processes that actually were present in reality. He had already begun to theorize about what was needed in order for a neural network to think, if you like, or become useful. And he said, look, what you need is a type of synapse such that if the postsynaptic neuron is firing when the presynaptic neuron is releasing the neurotransmitter, in other words, when the presynaptic neuron is firing, then the synaptic efficiency is increased. If you get that, then he could show mathematically that you have a neural network that can be useful and can process information. And what we mean by synaptic efficiency is the likelihood of being stimulated into further firing. So when we talk about synaptic efficiency, we talk about increasing the strength of a connection. These are the same things. Okay, we're just talking about them in different ways. So that's what we're looking for now. We're looking for what we call a Hebbian synapse. Because we haven't got a biological mechanism for it. All we've got is Donald Hebb in 49 saying, this is what we're looking for. This is what we need to find. And if you put that type of Hebbian synapse into an artificial neural network, you can show that it can learn. And I've already mentioned, we've got human-like learning curves, human-like errors. It can do pattern recognition. It can make best guesses. It can develop cognitive maps. It gracefully degrades. So that if you take out a neuron, it doesn't work so well, but it still works. If you take out a part of your computer code, nothing happens, just crashes. So we still need a biological mechanism by which neurons can change their synaptic efficiency when adjacent neurons increase their activity. We are looking for a Hebbian synapse. And so what we're looking for is a type of biological process. And it was found by Bliss and Lomo in 1973 in the rabbit hippocampus. And it's called long-term potentiation, an enduring increase in the size of the postsynaptic potential when you're stimulating a neuron. What I do, we now know it's actually common throughout the brain. He found it in the human brain as well. We found it in the rabbit brain, but it's common throughout the brain. What I love, I've got to share this quote with you that I found. As for me, it was certainly not prior interest in possible memory mechanisms that led me to a discovery that was in some ways accidental and in other ways the result of intuition that has often, I feel, brought me to look for or see phenomena that turn out to be new and interesting. And I love that because there's always this sense in science that we're supposed to know what we're doing, But actually, there are so many. When you look at so many of the great discoveries, like X-rays, for example, you would never have got a grant for. These are accidental things, and usually, I've heard it said that the phrase "oh, that's weird" is usually the beginning of a scientific discovery. It's when things don't happen the way you expect them to. But you always have to pretend when you're applying for a grant or something that you know exactly what's going to happen, which is ridiculous. But there you go. So this is the sort of thing that was done. You've got a stimulus on one part of the rabbit hippocampus, and you're recording the output in another. And you're actually measuring the excitatory postsynaptic potential, which just means that the firing that's coming out of the neuron that's after the neuron you're stimulating. Okay, so the excitatory postsynaptic potential is being measured, and it's already firing a little bit. There's always some baseline firing. And then you put in your stimulus, here it's called a tetanus, and of course you see an increase when you're putting in that stimulus. Well, that's not surprising. You're putting something in, you're getting something out. Not a big deal, really. A conductor would do that, a piece of wire. But what actually happens after that is that the baseline firing rate, if you like, of the neuron appears to be increasing for about 3 to 60 minutes later. So up to an hour later, that neuron is still firing more than it was before. So it appears to be responding. Now, of course, I'm not saying that that's necessarily a Hebbian synapse. I mean, there are different types here. There are non-Hebbian long-term potentiation, which doesn't require simultaneous depolarization. In other words, if you're just putting in some extra stimulus here, you find afterwards that it's firing more. A Hebbian synapse is when you're putting in stimulus here and it's already firing, and then you find an increase in the efficiency as a result. So we are still looking in more detail for a coincidence detector. And we find it in this explanation, which is the more modern understanding. And it provides a more sort of detailed account of how long-term potentiation occurs. And you might say, well, what's all this got to do with education? But actually, what I'm explaining now is you need to understand, for example, if you want to understand cognitive enhancing drugs. So this is like a new wave of issues that's entering education. Now we have drugs that can enhance cognition, and students are using them. In some universities in America, up to a third of students are using things like Ritalin to improve their exams. Well, you need to understand these processes to understand how those drugs are working. So long-term potentiation depends on a postsynaptic glutamate receptor, a particular type of receptor called NMDA. Now, normally, when you're looking at when you're looking at a synapse, this excitatory postsynaptic potential, in other words, what's coming out on the other side, is mediated by AMPA receptors here, which is another type of glutamate receptor. And these NMDA receptors, which I'm saying are the important things for long-term potentiation, are actually blocked with magnesium ions here. But when you get sufficient incoming glutamate to generate enough output here, actually it depolarizes this membrane sufficiently for the magnesium ions to become dislodged. And suddenly that means there's an extra way in which activity here can be passed through to activity here. In other words, if you've got enough activity going on here that this is depolarized, then this opens up as well as that. And that means that activity here and activity here produces an increase in the overall efficiency of this connection. In other words, you have your coincidence detector. You have your Hebbian synapse. Because of that, we have a basis now for learning in long-term potentiation. Now, unfortunately, that process only lasts a few hours, maybe weeks. And we know that, and we hope that, learning lasts a little bit longer than that. So we need to be finding some other, what we call late long-term potentiation. Late phase long-term potentiation to explain what happens later. So I've just explained early phase long-term potentiation, but we need something that happens later for learning to really be fixed in the brain. In the late phase of long-term potentiation, we think that the calcium enters the cell and triggers calcium-modulated protein. And that produces a chain of events involving the nucleus of the cell, producing protein synthesis and structural changes. In other words, the formation of a new synapse. So this is a lot more serious in a way, because yes, we can have early phase long-term potentiation here, but eventually, because of this calcium interacting with the nucleus of the cell, we have a generation of new proteins which can produce new structures. And in particular, we're talking about a new synapse, an entirely new connection forming. But that's not going to happen immediately. We need early long-term potentiation, and then we have this later long-term potentiation in which an actual synapse is formed, a new synapse. Now, do we know that this is the basis of how we learn in classrooms? Well, you will often hear neuroscientists talking as if we do absolutely know this, but actually all the evidence is circumstantial. I mean, if you ask the average neuroscientist on the street, how do we learn? He will say, or she will say, long-term potentiation, early and late, very important. That's what it's all about, okay? So it's kind of like an accepted dogma, and I do accept it myself. But just be aware of the fact that when you hear people in, teachers or people selling you neuroscience in education saying, oh yeah, we all know that connections in the brain are really important for learning. Well, actually, most of what we know about learning in terms of making connections between concepts is from psychology. We haven't got the technology to see changes in synapses as a result of classroom learning, okay? The only way we could do it would be, well, post-mortem, okay, which clearly is not ethical. But, it's all fairly convincing what we have around. I mean, this is some of the circumstantial evidence. We know that short-term memory becomes long-term memory only if you have this protein synthesis to maintain that synaptic enhancement. If you don't have that protein synthesis, if you don't have the ability to make those new synapses, then it's not going to happen. We also know that mice that are treated with NMDA receptor blockers, which actually stop that early long-term potentiation, can't find their way around a water maze. They can't learn, they can't get that cognitive map that they need. And we also know that rats, when they're conditioned to freeze to a neutral sound, the synaptic potentials from the amygdala become enhanced only while the learned response continues. In other words, once they forget in terms of what we understand from or what we assume to be their NMDA receptors, then the learning stops. So if you block the receptors, the learning doesn't happen. But there are other mechanisms involved with learning. One of them is long-term depression. So in other words, we have a reduction in firing rates, for example, in the perirhinal cortex, which is involved with face recognition. So you learn face recognition by a reduction. It involves a reduction in firing rates. And we also know that there are other processes which we don't quite understand, and one of them is neurogenesis. We do, now we know, generate new neurons, particularly in the hippocampus. The hippocampus is a very important structure for memory, encoding memory. However, we don't really know what that means in terms of learning. I mean, it's probably, common sense tells you that's going to be really good news, and actually, for those of us who thought we just had a diminishing, stock of neurons, I've often thought that I must surely have lost most of mine, It's really nice to know that you can produce new ones. And I sat through a wonderful presentation by somebody who's really at the forefront of this work lately. And she was telling us the sorts of things that are linked to this generation of new neurons. So this is a new area, if you like, for neuroscience and education to be looking at. It's a bit torturous, I have to say, what you have to do. But if you really want to generate some new neurons, you want to run around, make yourself hungry, and do some really difficult maths. Okay, I can't remember the exact order that you have to do those things in, but I've got a feeling that going for a run before lunch and doing some difficult maths is a really good way of generating some new neurons. No point doing it after lunch. Something to do with Guerin in the stomach. It's beyond me. Anyway, so we have now all the information you need in terms of understanding learning at a cellular level. We know that if we have networks of neurons, and we do have networks of neurons in which the efficiencies can change, particularly in a way in which depends on there being a coincidence of a neuron firing there when the neuron connected to it is also firing, then that network can adjust itself and indulge in intelligent behavior, intelligent learning behavior. Maybe I shouldn't use the word intelligent, but helpful, useful learning behavior. So now we could sort of move out a little bit and say, okay, so where in the brain is this occurring? When we think about learning, and I'm going to talk about learning now in terms of memory, most of what we know about, or most of what we have known about learning and memory for many years has been from psychology, and psychologists have enjoyed categorizing memory in different ways. So one basic division is between declarative and non-declarative. Things that we can talk about, like facts and events, and things that you can learn without necessarily being able to talk about how you do it, like riding a bicycle. But also, reflexes, some types of conditioning, all sorts of things. Priming. Priming is when you respond differently to one event because of a previous event. And you may not even know how you do that, but it will happen. And of course, we have these. We have many other ones as well, like explicit, implicit, memory and habit, semantic, episodic. Actually, these categories haven't been terribly easy to find in the brain. So if you look at different types of memory in a neuroimaging scanner, you don't tend to find that memory is organized terribly well in terms of these ways. So these divisions aren't necessarily that helpful for you in terms of understanding memory in the brain, I'm afraid. And this, of course, is what happens, because psychology has been around for a long while, neuroscience is new, neuroscience is making us think again about these mental models that we have about things. One division that does seem to make sense, though, is this declarative and non-declarative memory division. That does seem to really help in the brain. After that, facts and events, pretty much, both of those are to do with medial temporal lobe, but some other regions as well. And then non-declarative memory, procedural skills and habits, things like reinforcement learning, how we learn to forage, because we forage as well on a beach, humans forage. We forage on the internet when we're looking for information, stuff like that. That tends to be about the striatum. Emotional responses tend to do with the amygdala. Some skeletal responses tend to be to do with cerebellum. So you can very, broadly try to locate some types of memory in different regions. Some of these divisions do make sense. But I mean, even with reinforcement learning, for example, that actually, in terms of understanding reinforcement learning, it involves the cortex as well, and it can involve the medial temporal lobe in terms of episodic contextual issues and things like that. So it's not very clear cut, but certainly declarative and non-declarative is a useful division. So this sort of gives you an idea of how distributed a memory might be. So if you say to somebody, do you remember when we walked across the Clifton Suspension Bridge? Well, yes, I do. And now it comes to mind, what I'm actually doing is retrieving a lot of information maybe about the location, which has been linked to parietal lobe activity, information about the shape, which has been linked to inferior temporal lobe activity, information about color, temporal occipital, areas, but also a lot of interaction between these areas, and then you're actually drawing all that into your conscious attention, which means you're going to be using a lot of dorsal lateral prefrontal cortex probably as well. So just when you say to somebody, oh, remember when we walked across the Clifton Suspension Bridge? Activity all over the brain. So, however we categorize different types of memory, a simple word can stimulate so many different areas of the brain at once. And so it's really good to think about a memory as distributed across many different brain regions, some more associated with particular aspects of a memory than others. I've mentioned working memory. So how does working memory get dealt with in the brain? Well, it's a bit of a mystery, actually, and we're still trying to sort this out. But an interesting experiment that has shed some light on it suggests that working memory may just be a persistence in neuronal firing. Okay, so you have the stimulus, and that stimulates the neuron, but actually it carries on. Maybe it's a bit of an early long-term potentiation or whatever. But we can see this in an experiment that was done with monkeys, whereby a visual, they were looking forward, and then there's a visual cue here, and essentially they have to remember where that visual cue was in order to get the piece of food or whatever. And we find these are all the correct trials. And when we look at the neuronal firing, the firing of a neuron associated with working memory for that particular task, for all the correct trials, we find that that carries on firing right up and sometimes beyond the time at which they make the response. Even this one here, at least it carries on right up until they make the response. Whereas for the incorrect trials, the times when they forget where it was, we find that the firing of that neuron actually desists some period of time before the response is given. Okay, so in other words, it's fled from working memory, it's fled from their consciousness. Interesting experiment. I think it's important, this was like done with one neuron, but it's important to remember that working memory is also widely distributed across the brain. We tend to associate it most with the dorsolateral prefrontal cortex, but it is quite likely that working memory type effects are present in a wide variety of different regions distributed across the brain as well. Dorsolateral prefrontal cortex, and you know where that is now, don't you? Because you know about dorsal, you know about prefrontal, you know about what lateral means. Oh dear, there's one there which I didn't tell you about, orbitofrontal, just underneath the front there. Very difficult bit to pick up on an MRI scanner because it's so close to the sinuses. The sinuses cause all sorts of distortion. Okay, so are there any questions on that? Because I've got to go on to a little bit of evolutionary psychology. I'm just checking the time. Yes.
The reason I have this that you mentioned through the later stage of LPP, do they last forever? Do they stay there?
Oh, well, that's a good question. Of course, we don't know in those particular experiments because, sadly, I mean, they're done usually on animals. Well, I imagine always on animals, and the animal's destroyed in order to be able to study the synapse. So it's not as if we have a chance to carry on looking. I should mention, by the way, this is going out publicly, I do no experimentation on animals before I get a bomb. So yeah, so that's, so we don't really know. What we do know, for example, is that there was a wonderful study done of Carmelite nuns where they showed that, those nuns who had indulged in new learning activities had much greater synaptic density, many more synapses. We also know that synapse density is related to education as well. Although somebody at Bristol here is doing some work on that and has been coming up with some interesting data which tends to suggest it doesn't necessarily mean you have more synapses in some regions of the brain. So clearly, early experience has an impact on one way or another, so it does carry on for years. But I mean, I guess the other way of looking at it is that we know that memories can carry on for years and years. So I suppose, unless they're reconstructed in some way, must be the case. Well, you see, that presumably, just to repeat for the camera, someone's asked, presumably some die off because we don't use them. And that presumption is made by all neuroscientists. But actually, I'm not sure it's ever been tested. But you're right, that's another one of those basic assumptions. That if you ask your neuroscientist in the street, they will say, yeah, synaptic pruning, because that's a developmental phase that occurs during development. So, right up until the age of 19, we are, encountering waves of synaptic pruning when the number of synapses we have is actually being reduced. Now, we assume that that's because they're not being used, because it makes sense, doesn't it? It's like increasing the efficiency of what you've got, There's no point in powering synapses that don't do anything. But in terms of finding the evidence for that and saying, look, this person hasn't used their synapse and it's gone, I mean, I suppose you can go back to the Carmelite nun study, and certainly, there are a lot of studies that have been done on how you've had, relationships between synaptic density and learning. But yeah, I think it's a good presumption. It's a good assumption. Yes.
So LTP happens when the two neurons are firing at the same time.
Well, there are that's the one that we're most interested in. So that's the type of that's the type of LTP that we are most interested in. But I think synapses can change their efficiency in other ways. Okay. But yes.
If you explain why it would happen that they weren't firing at the same time, is it because it's already sent an action potential, so it's just firing quicker, or is it amplified by a different neuron?
Well, if you go back to conditioned stimulus and the sausages are arriving in the bowl at the same time as the trumpet is hooting, then it's helpful to have an association between those two events. So you can see that coincidence detection at that basic level can actually produce simple associations like that. That's fairly straightforward. But actually, that type of Hebbian synapse can do more complicated things than that. I mean, actually, how they do these things is a little bit difficult to explain, and that's one of the problems with neural networks. We can model these neural networks whereby we have like massive layers of artificial synapses, and we can see them doing quite intelligent things. And then, of course, we have to ask ourselves, "Oh, that's very clever." You can tinker with the learning rules, and you can tinker with the efficiency and the rate at which they learn. But then, when you actually say, "Oh, it's done something really clever," now how did it do that? What you're faced with is massive numbers of matrices, matrices with massive amounts of numbers in them, and a very messy type of, what appears to be a very messy type of solution, nothing like a nice clean flow chart. That's why we like nice clean serial flow charts, because we can explain them to each other. But the way in which parallel networks learn is very messy, contains a lot of redundancy. It's very intelligent, but when it comes to actually looking at it and saying, you see this part of the network is working that bit out, and then this part of the network is working that bit out, doesn't tend to work like that. And this is one of the problems because the psychologists have for a number of years loved their little flow charts, the Badley and Hitch model of working memory, for example. Actually, the brain doesn't work like that. When we think about reading models, we think about decoding phonologically, we think about then linking it to meaning, maybe looking in our lexicon of words. And we think about these things in a serial way, because it's easier to think about, and that's how we read a book, isn't it? But actually, when I say that's how we read a book, I mean, we start at the first page, we go to the last page. It's a sequence. But actually, when you look at the brain operating, the evidence tends to suggest that all these areas are very interconnected. So, yeah, so it's a bit messy, basically. Yes.
Well, I think I'm sort of asking the same question. I haven't quite got it. Something, if you've got a neuron further on along the network, something must trigger that. From the saying they fire at the same time. Something must apply first, whether it's this neuron or not, because this one's fired.
Yeah, I mean, maybe I could explain this by saying how we train artificial neural networks. So if you're trying to train this network, let's say you want it to recognize that a particular letter is a T is a T sound, so in order to train this, what you do is actually clamp on to the output a T sound and then keep presenting T's here. Okay, so if you're trying to make a link you're trying to train your artificial network to intelligently link letters to sounds, then you keep clamping on the sounds here and putting in the letters, the correct letters here. Okay, and eventually, if you've got coincidence detectors, if the synapses are adjusting themselves, as Hebbian synapses, the efficiencies of these connections will change in such a way that eventually you can take off your training corpus, your sounds, and when you add a letter here, it will automatically output it as a sound. And that it can only do that because it's making a relationship between what you want it to do here, the output, and the input there. And that's why you need coincidence detectors. That's why you need Hebbian synapses. Things that actually increase their efficiency when that's firing and that's firing.
But for the output to fire, if the input fires, something in the middle would have to fire for the output to. Does that make sense? Like it comes away through the network.
Yeah, so we've got axons going through. So let's just say we set all our efficiencies to 0.5. Okay, so you get 50% or whatever. So if you put something on there, then you're going to get something out there, but it's not going to be meaningful. What you want is for these efficiencies to adjust themselves so that you only get what you want here when you put in the right thing there. Yeah? So you need to, these synapses need to adjust themselves so that the efficiency, they need to adjust themselves according to the relation between the input and the output. So they need to take on board the fact that both of those things are firing before they increase their efficiency. That's the Hebbian synapse, basis of the Hebbian synapse. Yeah.
So you mentioned before that our brain can create new synapses.
Yes.
It's happened throughout our lives because I have.
Yes. No, actually, synapses, yes, all throughout your life. There's no problem with that at all. The amazing thing is the neurons themselves are actually regenerating to some extent. So we used to think, so up until like about 4 or 5 years ago, during the development lecture, which you'll have next week, I used to say all of your life stock of neurons are produced within the first 3 months of your conception. Now that's sort of 99% true still. But now we've found out that even after you're born, you can actually produce new neurons, particularly in the hippocampus, which is very important for encoding memory. And there are particular conditions that encourage that to happen. And one of them is related to this Gerin hormone, which is beyond my understanding. But being in a particular state of appetite, challenging yourself with a mental task, and exercise are things which are linked to this generation of new neurons. So you're right. We used to think that you had all your neurons well before you were born, and 99% of them, that is true. Synapses, on the other hand, which are the connections between the neurons, you are making and breaking those all the time. That happens throughout your life in a big way. And actually, the evidence is that if you want to make lots of synapses, challenge yourself with new mental tasks. Okay, it's not enough to carry on doing the crossword. Try chess, or if you do chess, try crossword, whatever. And there's also these brain training games and all sorts of fun things. Yeah.
So when you get your new synapses, are they specific to the activity or learning you're doing when you got them, or can you then use them for other stuff?
Oh, very good question. Are the new synapses when they arrive of general use? I don't think that they are of general use, but we don't really know. I mean, for them to be useful, they have to respond to the things that you've learned. So therefore, we would expect them to be learning specific. However, it is the hope of many people that there are general processing systems in the brain, such as attentional systems, that if we can improve those, and working memory systems, if we can improve those, then that will be useful for all learning. So there have been many attempts at, I'm not sure if we're going to, we'll deal with this in issues in neuroscience and education. We haven't got much time to deal with it in this unit, unfortunately. But working memory training is something which appears to show some promise. So Torgel Klimberg has been working in Sweden, has been working on really interesting work whereby children appear to be able to increase their working memory as a result of computer-based training. And this is really hopeful because working memory is very closely related to how well you do at school. Actually, I think it's one of the best predictors we have of that. So there may be some general processes whereby if we practice them enough and we increase the synaptic efficiency or whatever, then it's not just increasing synaptic efficiency. We selectively learn and we develop our neural networks so that the synaptic efficiencies are appropriate. Then we maybe get good at a whole range of different things. But actually, I have to say, beyond working memory, scientists have had real trouble with that. What is of interest is that action video games appear much more successful. So action video games appear particularly good at increasing synaptic plasticity. So good, in fact, that they are being used for interventions with trauma in visual processing systems. So if you've had an accident and you've damaged part of your visual networks in the brain, action video games are being used to improve the rate at which you repair yourself, the brain repairs itself. Yes?
I'm across an article, I think it's very good. It's about rats getting new synapses in the cerebellar cortex when they learn new acrobatics.
Yes.
Whereas the ones that were just exercising wouldn't gain new synapses. So, but it was just in the cerebellar cortex.
Okay, so just to repeat for the camera, someone's talking about a study in which rats have been shown to increase their synapses or their neurons?
You talk about synaptogenesis.
Okay, so the rate at which they're increasing synapses in the cerebellum as a result of exercise. The cerebellum traditionally has been associated with more sensory motor processing. I mean, actually, we're now thinking it's related to a lot more than that, including reading, for example. But it's not a surprise, I guess, therefore, that if exercise increases synaptogenesis, and there is evidence to suggest that it does, that synaptogenesis, that making of new synapses, occurs strongly in the cerebellum. If all those rats were doing was exercise. But it's been shown in humans that exercise can improve short, medium, and long-term memory for information that you receive afterwards. And that's certainly not just the cerebellum. And we assume that that's related to, well, it would have to be related to changes in synaptic efficiencies, synaptogenesis for long-term memory. So I'm glad that you're asking me these questions because it makes it, it gives me an opportunity to show that this is actually very relevant to learning. And this morning, the Wellcome Foundation and the Education Endowment Foundation are announcing 6 projects for neuroscience and education in schools that's going to involve 70,000 children. And one of them is going to be the introduction of gaming, teaching science entirely through gaming in year 8, which I'm coordinating from this department here with Manchester Metropolitan University. But another one of them is exercise, and using exercise to improve academic achievement in schools. So it's very relevant, really, to what you're discussing. Any other questions? Well, we can have more at the end, so don't worry. Okay. Oh, I went back a bit, didn't I? Okay, yeah, so I'm going to move on to evolutionary psychology. I used to love doing these slides, and now when I do them, I think, oh, I'm not so sure about this, really. Okay, so evolutionary and species perspectives. Our learning mechanisms have evolved. Yes, they have evolved. Have they evolved? See, I'm getting very skeptical. Yes, they have evolved. This provides insights at a functional backwards design level if we think of evolution as a designer. Okay, well, what's the idea here? Well, if you think about evolution as a designer, which is a dodgy thing to think about in the first place, but if we do think about it in that way, then we can think that our prehistoric environment, whatever it was, obviously can be associated with particular needs that we had in order to survive. So we can look at our prehistoric environment and say, well, so, actually we had to get on with a lot of people. Probably keeping the tribe together was quite important. Getting on with knowing who your kith and kin are, knowing who your brother and sister is probably quite important. Knowing that that person is not in your tribe, that's probably quite important too. So, by thinking about the environment, you can think about what we need to learn, okay? And then you can think, well, okay, that must have influenced the way in which the brain works. So we should probably, after all those hundreds of thousands of years of evolution, we should probably be looking in the brain for mechanisms that are particularly dedicated to recognizing your kindred, recognizing people that you know, your family, etc. And we look for those principles, and we should therefore also be able to see those principles at work in the product. So I should be able to see that you're much better at learning to recognize your friends than you are learning genetics or something which prehistoric people did not have to learn. Okay, kind of makes sense. So if we understand the prehistoric issues and the specification of what we needed to survive, then that might help us understand the design that underlines what we've got now in terms of our behavior and what we see happening in the brain. So, was there a specification? We have to ask ourselves. Every time we see a particular type of behavior, we can say, well, if this is a real behavior that we're looking at, if this is something which we think, is significant, and if we think we have a theory underlying what's going on, is there a specification in terms of the needs, the prehistoric needs, that would actually have produced that? Okay, so for example, we have a love of sugar. Yeah. Okay, we appear to be very dedicated to seeking out sweet things. They rot our teeth. So why? Why do we do that? So is there any sort of specification back here, in our prehistoric times, that would justify us seeking out sweet things? Well, of course there is, because back then, before sweets were invented and sugary drinks that rotted our teeth, sweetness meant honey, it meant berries, it meant fruit, things which are actually very good for us. And so sometimes going back and looking in the past can help understand why we behave in particular ways, even though that is a maladaptive trait now, because if we eat lots of sweet things, our teeth fall out. Back then, it was probably a very adaptive trait. It did actually help us fit the environment that we were in. Okay, so evolutionary perspectives are probably quite important, but I think one does need to look beyond just the human species and look at other species as well. My problem with this is that, in fact, evolutionary psychologists do tend to focus on the Stone Age. And what we understand of the rate at which evolution has moved is that actually, really we're looking at a lot of behaviors where there would have been no evolution. So in terms of reading, in terms of mathematics, our brain has not evolved to do those things. And actually, the messages that the strongest messages we have about understanding our brain through evolution is by looking at a much deeper time frame, understanding how other species operate. And so, I would say if you really want to use evolution to understand the human brain, you should be understanding and respecting the complexities and the sophistications of the way other animals think. Because we share many of those systems. And the more you get into neuroanatomy and the more you understand the brains of other animals, the more you begin to realize that we are not some sort of outlier in evolution that has arrived here as a result of what happened in the Stone Age. No. No, the skills and abilities that we have now are those that you would expect of a typical primate of our size. What makes us different to the animals is actually the culture that we've developed, which is something which has, really gone ahead of our genetic basis. Okay, we've not genetically adapted very much as a result of our culture, not in terms of our cognition anyway. So I have got quite a few criticisms of this approach, but there are a few simple things which are worth bearing in mind. You might ask yourself, for example, conditioned instrumental slow learning, why do we have it? Why does it take so long for you to learn? I could ask you, are you still struggling with understanding long-term potentiation? Why is that? Well, actually, there's a good reason for it, because if you made connections, synapses, very, very quickly, you could end up in all sorts of messes by assuming that everything that's happened once is going to happen again and again and again and again and again. Okay. So it does make sense that we need to have time. There should be a time delay in us learning some of these special things, because otherwise we'll just overload. Actually, an interesting example of that in a way is Luria's S. This was a guy that a Russian psychologist, Luria, I think he's Russian, forgive me if he's not Russian, studied. And he had a fantastic memory, so that he was actually read a Latin poem, and when in his early life, and he's only ever read it once, and he could remember the whole thing like 20 or 30 years later. Incredible memory, bizarre memory, but it also meant that he was permanently distracted. And if you think about it, if you could remember every instant of your life, everything that's ever happened to you in vivid detail, you wouldn't be paying attention to me now, would you? You'd be in there somewhere, enjoying it again. Okay, so there are problems, therefore, if we suddenly make connections that we can, that are there straight away. And then there are other types of learning that we see which actually do occur just once. And one of those is the Garcia effect. Have you ever been ill after eating a certain type of food or drinking a certain drink, and you've never been able to touch it again? Right? I have trouble with mushroom risotto because of an incident. It wasn't the fault of the mushroom risotto. I was in a country where I shouldn't have been drinking the water. Okay, drank the water, had a mushroom risotto. I won't tell you what happened next, but the fact is I've never been able to eat it again. This is called the Garcia effect. It's a rare example of one-off learning, one experience, and you never ever forget it and can never go back to it. And of course we think that was that's a preparedness that you're born with for a particular reason, and it protected us probably back in the old Stone Age, or environment, well, beyond that actually, because well beyond the Stone Age, because if something is poisonous and you've thrown up on it, you shouldn't ever go near it again. Right? You may not get a second chance, and so the body prepares you to the body is prepared to forever avoid that food, even if you know up here that's not the case. It's other parts of the brain. It's not a higher order thinking process. It's a conditioned response, and it will prevent you from ever eating that food again, or at least for a long time. And then there are other things where we have what appear to be species-specific preparedness, or at least I'm not sure if it's a species-specific knowledge, but at least it's a species-specific preparedness. Some people would say it is knowledge to avoid certain other types of species. So there are examples where they've shown certain types of animal appear to know that when they're on the ground, even as babies, they know that a particular type of shadow in the sky means danger. Even though they haven't never been taught that, they're born with that awareness, they're born with that knowledge, and that makes sense as well. But at least we have, at least we often have at the very least, we ourselves have a preparedness for knowledge. So if you are one of those people like me who really are not keen on these creatures, I can't explain why that is, because I know that all the spiders in this country are not harmful. I do not live in a country full of harmful spiders. But if that thing walked over my desk, I would be right out that door immediately. I can't cope with them. Okay, so why is that? Well, it's because you know, we have evolved through a long period of time an awareness that, not an awareness, but it's actually, you know, we're born with a preparedness to understand that a particular type of species is potentially lethal. And the same thing with snakes, of course, as well. So we do appear to have, if not knowledge, and that's an arguable one, but at least a preparedness for species-specific knowledge. Okay, so I wanted to sort of, I think I have gone slower though, haven't I? Yes, okay. I wanted to emphasize as well this idea about structure, because this is really a very impressive effect of education. And there are many students, I'm thinking really about children in schools, who feel that their brain is a limiting factor in what they can learn. You know, I can't learn it, miss, it's my brain. There's a sense in which the brain, many people have a sense that the brain is fixed, and this is really not the case. We've talked about the way in which connectivity changes, and if the connectivity changes, then it's not difficult for you to imagine that the function also changes. So when you're learning complex mathematics, there is a study that shows, this is in adults, that as you learn, so there is a decrease in activity in dorsolateral prefrontal cortex because you're using less working memory. As it becomes more automatic, you have to hold it less in your conscious attention. And there is an increase in activity in posterior, more automatic regions because it's becoming more automatic. So you see a shift in functional activity as a result of learning. We know that there is a change at the cellular level in terms of learning. And we also see changes in the structure of the brain. And the most famous study is Eleanor Maguire's study with taxi drivers. And what she showed was that London cabbies, there was a particular part of the hippocampus. Now we're talking about posterior. You know where the posterior is now? It's the back, isn't it? It's the back of the hippocampus. So it's around there. If it comes around again, I'll be able to show it to you. So it's around that part of the hippocampus, which in London taxi drivers is larger than the rest of the population on average. Yeah. More impressively than that, it increases its size with the number of years of taxi driving experience. So if you have had 20 years of taxi driving experience, you have a particularly large posterior hippocampus. And that's because that's a region associated with visual spatial learning. Okay, so it kind of makes sense. Of course, there is this issue that the skull is finite, and therefore, if one part of the brain is increasing its size, stands to reason that some other parts of the brain must be decreasing their size. And which part of a taxi driver's brain is shrinking, I leave to your discussion. It's not species specific, we see it in animals as well. An excellent example are avian food storers like the scrub jay, who have an expanded hippocampus, usually in the wild, because they have to go across very wide ranges in order to find their food. They have to navigate across a very broad region. And yet, with the urban environments that have developed, we have communities of these birds that are now situated in cities where visual spatial navigation is not really an issue. And if you look at the hippocampus of urban scrub jays, you will find that they are much smaller because they just don't need to navigate across such wide regions. And one of the most impressive, because of the time scale that's involved, is a study by Bodran Dragansky, who asked people to learn to juggle. He identified regions of the brain that appeared to be activating as a result of juggling. You can see there's two here in the posterior part of the brain. We know we're looking at the back of the brain because there's a cerebellum, okay? And they are perceptual regions of the brain, occipital, moving towards parietal lobes. And what we find is that those parts of the brain, after 3 months of training, increase their size. When the trainee jugglers stop training, we find that those regions decrease their size. Not entirely back to where they started, but have decreased their size once they stop practicing. So even over 3 months, parts of the brain can change their size. Now, what is actually going on there, we don't know, because again, we can't do post-mortem studies in this way. We can't actually go inside the brain and look to see what is causing the increase in the size of that part of the brain. It's probably synaptic density, probably more synapses, very, very likely, but we don't know. Okay, so here we are. These are the things that we have learned today, I hope. I'm going back to the review. Oh, I've got to get off there before I fall off. We're going back to the review. Learning is often synonymous with memory and cognitive neuroscience, and in biological terms, change, another word for that is plasticity, in brain structure, connectivity, and activity can be called, in biological terms, learning, including decreases. All animals learn, and useful learning can be achieved by quite small networks when they change the efficiency of their connections appropriately. And one of the sort of synapse that can do that quite well is the Hebbian synapse. And that requires a coincidence of activation output and activation input. And then there is a change in the efficiency, i.e. responding simultaneously pre-post excitation. Biological connections between neurons are called synapses. Actually, I should probably go at point 2, it'd be better, wouldn't it? Biological connections between neurons are called synapses. There are electrical synapses, we haven't dealt with those. They're not really of great interest in cognition. Chemical synapses, the ones that we're particularly interested in. We've got electricity coming down the axon, but when it goes across the connection, we're actually looking at a chemical process. Early and late processes of long-term potentiation we've discussed. The early processes in terms of the magnesium ions coming out of those NMDA receptors and allowing a new way in which activity can cross the synapse. And it only happens when you've got pre and post excitation. And also late long-term potentiation, which we know is about calcium proteins increasing synapses themselves, the actual generating new synapses. Not just an increase in the efficiency of a synapse, but a new synapse. We also know that working memory may be due to persistent firing, persistent neuronal firing, just carrying on after the stimulus has gone away. Memory and working memory are distributed in the brain. There's not one little box where it all happens. Evolutionary psychology is another perspective on how, why, what we learn, which I'm a little bit skeptical of the way it's been taken. I haven't really explained that, maybe I should. Okay, so if you look at the evolutionary psychology literature, you will find a lot of other stuff about the fact that we have a module in the brain for identifying flowers, because that must have been very important in prehistoric times. We have a module in the brain for making tools, because that must have been very important in prehistoric times. We have a module in the brain for physics, because it must have been very important to be able to judge the distance when you're throwing a stone at an animal or some such thing. It must have been very important, that's right, it must have a module in the brain for identifying people who we're related to so that we can avoid incest, because that would obviously have depleted the genetic diversity. The trouble with this is that actually everything that we've just talked about in this lecture, I hope has convinced you that the cortex is really plastic, actually. I think it makes sense to talk about the human brain and actually the primate brain as being very suitable for socialization. But that's not because of prehistoric times, that's because we're primates. Okay, that's got nothing to do with the Stone Age. yes, I think we do have some modularity in the brain, which is very suitable and designed for socialization to some extent. But no, I don't think that's got very much to do with the Stone Age. In actual fact, the cortex is very general purpose. If you knock out one bit, another part of the cortex will take over that function. So I think in terms of understanding the role of evolution, we need to be thinking about understanding other species, understanding the role of the basal ganglia, for example, which is something that arrived with the first fish that have a jaw. Anyway, I won't go into that. I'm writing a book about that, except I'm not writing it because I'm never finding the time, but I could get too excited, so I shall hold back. Neuroplasticity. When we learn, it is not just connectivity and function that changes in the brain, but the structure changes also. Okay, so those are the main points that I wanted to get across. It may well be, I'm going to run across some of these in the next lecture. So we have a developmental, no we don't, we have an emotional lecture coming up, don't we? We have a lecture on emotion coming up next. For that lecture, I would be very interested if we have, do we have two friends in the audience? Maybe you two. Oh, go on. Yes, go on. Would you be prepared to participate in an experiment that will be publicly broadcast? I'll get back on my chair now. Would you be prepared to do that? Okay. You probably have to sign a form and everything to make sure it's all right. There'll be a. Okay, there might be a. Okay, we'll discuss it. If you can see me at the end, and then so that we can set that up. Thank you for doing that. I'm sure everybody will benefit. Actually requires me getting some technology working, which IT systems are having some trouble with at the moment, but we'll see how it goes. Next week, then, we're going to be having a little bit of revision on what we've done this week and what we've done last week as part of the emotion lecture, which I hope that you will enjoy. But if you want to ask me questions now about this or anything that we've done last week, now is a good time to do it. So it's all clear as mud. Yes. Yeah. Yeah. Yeah.
Is that like working in different contexts, or are they getting better at the kind of what they're doing to get better at?
It's well, I mean, you've hit upon the number one question, which is transfer.
Yeah.
So I've had a question about working memory, and is it really possible to train working memory in a way that can transfer to different contexts? It seems to be possible in terms of providing the tasks are not too distant from what you've been trained on, but it's the distance which is the controversy. And there's a lot of excitement about this, and there's products are being sold, and a lot of schools are using them. But the point that I have to make is that nobody, no, that's not quite true actually, but there's very little evidence to show that this is transferred to school achievement. Okay, so what you can do is train up working memory, put them on another working memory task, and show that working memory has improved. And that's very exciting. But what hasn't been done properly, and perhaps this is just because the research hasn't been done to demonstrate it, because it is very difficult doing school-based research in the real world, is to show that that actually transfers to everyday tasks. Having said that, Sebastian Lapina in Argentina has recently published in PNAS work that does show actually computer-based cognitive training appears to have transferred to school achievement. And it's the first study that I know of, so it's very exciting. I think it's interesting. I assume, because I know that Sebastian really focuses on disadvantaged children, so I assume that the sample is disadvantaged children. And I think that might be important as well, because there are reasons to think that some of these interventions might be more effective amongst those children. So maybe that's why it hasn't been shown before. I don't know. But it's how far it transfers and how different the context has to be before it stops working or stops showing effect. It's still out there. Yeah.
Nice to do with this, but I was just thinking that there's some stuff about those students and that transfers to mathematical abilities. Is that not the same thing?
Sorry, you've heard that what transfers to mathematical abilities?
Learning to play music.
Oh, learning to play music.
Tests, but it's actually on the website.
Yeah. Well, I think if the thing is, there's all sorts of questions there about what it is that's actually having the effect. So if you're training a child to be organized enough to turn up for a lesson, practice, maintain their practice at home, take pride in what they're doing, and receive positive attention as a result of it, you think of all the different ways in which that could be affecting their achievement in other areas, which is not necessarily to do with an improvement in cognitive function. So we don't know. What I certainly can say, though, is that the Mozart effect doesn't have any, as I know it, convincing evidence for it. But that's a different issue, I know.
Yeah, well, what I've got specifically, I think dyslexic.
Ah, well, that's an interesting one. Okay, so there is some basis for considering that practice in decoding music, in other words, listening to music and hearing the rhythm and the frequency information, is a little bit like practicing phonological decoding, because both of those things require listening to sound and the timing of sound and making sense of it. And on that basis, there is a reason to think that at least that should be researched. And I know that Usha Goswami, for example, in Cambridge, is very interested in that theory of dyslexia, that in fact it's a potential problem with the decoding of very rapid speech rhythms.
But that would be a kind of transfer.
That would be a type of transfer, but I think I think more evidence is needed to clearly establish it. I can imagine what I'm saying is I can imagine how it might work, but we need more evidence to say that it actually does work. I mean, there are some things that you hear like brain gym, and I just can't see how that can work. You know what I mean? So you need to differentiate between the theory and also, so if you've got a reasonable theory, okay, that's a good starting point. Then you need to see the actual practice and see that it actually impacts on school achievement.
It was very specific as well to, I think this one, guitar, but it was also using this sort of children.
So it's motor perception skills. Yeah, I have to say that there's very little evidence to suggest that the training of motor perception skills can improve literacy, unfortunately. Okay, so we are ending a little bit early again. I'm feeling nervous about this. We can't keep ending lectures early, but I imagine that if there's no questions and you feel entirely happy, what sometimes happens is that people come up and they ask me afterwards a question because they don't want to ask it now, and it's usually a very, they don't want to look stupid, but actually if you don't understand it, there's a good chance that somebody else will not have understood it and they will benefit from you asking it. So can I ask that person who's really just waiting till everyone's gone to come up and ask me the question? Ask me it now, go on. Go on, it's you. Okay. Well, we'll see. Okay, well, thank you very much for coming along. This lecture is now on YouTube, or it will be in 5 minutes. I think it takes 5 minutes for it to transfer across. And next week, back to the normal time, in the normal place, where we'll be having an exciting lecture, hopefully with a live demonstration of emotionality from our two kind volunteers over here. Okay, thank you.